The wetland lab that we're doing in ecology in our ecology section is fundamentally about ecology. And ecology, if you look at it in its most basic form, is looking at the distribution and abundance of species. So where are species? Why are they in certain locations? How many of them are there? And all the interactions that play into the distribution and abundance of species. In this picture, you can clearly see that there is a boundary in between the organisms that live here. So if we were to identify that boundary would exist right here. So there is some reason why, if you look at this section right here, section A, and if you look at section B, those two areas are very, very different. So the question that ecologists would answer, or want to answer, or to ask would be, why are the different species and the distribution and abundance of the species different in these two areas, in location A and B? And fundamentally, that is the question that we will be asking in our wetland study. This is an aerial photograph of Milton Academy from Google Earth. And just to get us oriented a little bit, this is the ACC. This is the track. Lake O'Hare is right here. And this is the quad. When we go to the wetland that we're going to study, we're going to come down Center Street and we're going to travel down Randolph Avenue and we're going to come into the wetland right about here. And if you look at it, the area that we are going to be studying is this area located by that circle. So this is a zoomed in picture. This is the Lake O'Hare. And we entered in we're going to enter into the wetland somewhere around here. So in the last slide we talked about the distribution and abundance of species. If you look at this picture, you can clearly see that there are two areas in this picture, or two distinct areas in the area that we're going to be studying. One of them is here, and that area is the forest section. And if you were to take a ground level shot of the forest section, it would look something like that. And the other one here is the wetland, and the wetland ground level shot would look something like that. What we're going to be looking at is the boundary that exists between these two areas, between the forest and the wetland, runs right here. So the question that we're asking is, why is there a difference in the distribution and abundance of species between the wetland and the forest? And to do that, we're going to do something called a transect study. If we zoom into the boundary that we'll be focusing our attention on, we can see it here where the wetland section is over on the left side and the forest is over on the right side. We're going to use a sampling method called the transect with quadrats. What a sampling method is a way of looking at an ecosystem and looking at the distribution and abundance of species in that ecosystem. We could spend the time and look at the entire area and count all the species that live there, but that would be too time consuming. So we employ this sampling method. What this is a very basic and easy way to be able to sample the species that live here. The first thing we're going to do is we're going to run a transect that runs from the wetland over through the forest. And what the transect is essentially it's a line, and we'll make sure that it crosses the boundary that we're looking at. Again, that boundary is right along here. And we're gonna take a piece of rope and we're gonna string it from the forest over into the middle of the wetland, and that will give us a cross section in which we can study. What we will then do is we will look at 1 meter intervals along this transect, and these are not drawn to scale. And we will place what's called a quadrat down at those 1 meter sections. And a quadrat is in this case, it's going to be a 1 by 1 meter square, which we will count all the organisms that are living there, what organisms are there, and how many of each organism. And we will also look at various abiotic factors. And we can do a quadrat at each 1 meter interval as we're moving down the transect, and in that way we can get a representation of the organisms that are living in this area, how many of them are there, and what abiotic factors exist at those various locations.